everyone, and if you can hear me loud and clear, turn on your cameras and then wave your right hand so I will know you are here. Okay, if some of you still can't hear me, I don't see you waving or turning on your camera, but if you are okay, Omari, I see you using technology to wave, so which is fine. So today is our last day of this training series, and many of you have been with us from the beginning. So we are going to grace this with the final presentation from Dr. Adam Filippi. Dr. Filippi is a senior investigator and head of the Genome Informatics section of the National Human Genome Research Institute and Institute of NIH. His lab develops foundational methods for genomics, focusing on specifically on the problems of genome sequencing, assembly, and comparative genomics. He is a co-founder of Telomere to Telomere Consortium. I'm sure, Adam, maybe you can say a word on that one once I hand over to you. And as a co-founder, he is currently developing new methods for the complete and gapless assembly of human genomes using long-read sequencing technologies. His homepage can be found. I'll post his homepage in the chat area. So for those of you who want to check more on him and then what his lab does. So Adam, we thank you for accepting this invitation and then being with us this morning to give us this topic. The title is Finishing the Human Genome. So Adam, thank you again, and then it's all yours now.
Great, thank you. John, do you see the full screen or the presenter display?
Yes, I see it.
Is it filling your full screen?
No, it's not filling the full screen, but I'm able.
How about that?
Well, I'm able to do that to fill it. I don't know if everyone is able to do so.
I guess I'm asking, do you see the correct display or the presenter view?
Yes, I do.
Okay, good. Well, thank you, John. It's my pleasure to be here with you all this morning. And I understand that this is a data science crowd, and so I definitely will talk about this consortium that John mentioned in my intro. But given your interests and backgrounds, I'll also try to give it a personal touch and explain to you a little bit about my background and my journey through data science to where I am today. So a little biographical info for myself. I've been in and around Maryland for the last 20 years or so. I did my undergraduate in Baltimore at Loyola University, and I did my PhD work at University of Maryland a few years after that. And in between and after and around, I've been working at various research institutes and government agencies here in Maryland. And my training, both for undergraduate and graduate, were in computer science. And so it closely aligns with this theme of data science that you all have been learning about this spring. And data science is a relatively new term. It was called informatics when I was younger. Computer science, data science, I kind of view them all as the same. It's taking information using computers to analyze and better understand that data. And that's what I've been doing my whole career. Most recently, I'm at the National Institutes of Health over in Bethesda. I heard that you have had some presentations from some of my colleagues this spring. And all of the knowledge that you'll hear from me today on genomics and the human genome has really been on-the-job training. I haven't had a biology course since high school. All of my focus has been data science, computer science, and applications, most recently, to genomics. I think that's one of the most empowering things about a field like computer science or data science is that you can take your talents and apply it to any field that you find interesting or are curious about. And so I've found it really rewarding to apply my computer science skills to the field of genomics and human health. So today I'm going to talk about a little bit about the history of what the human reference genome is, and how we got there and why we're still working on finishing it up. So for those of you that are old enough to remember what happened 20 years ago, you might recall that the human genome was announced finished back in 2001 to great fanfare. And here is the announcement shown in the White House as announced by the current NIH director, Francis Collins, and the CEO of a commercial company called Celera Genomics at the time, Craig Venter, and Bill Clinton was president at the time. And they were calling it Decoding the Book of Life, a Milestone for Humanity. And it was published in the premier journals of Science and Nature, both the private effort and the public effort simultaneously, describing the first sequence of a human genome, where we have read the individual DNA bases from a few individuals, stitched that together into the sequence of the genome. And this was June 26, 2000. The issue is that it wasn't actually finished back then. They finished the majority of the genome, say 90%, but there was still 10% of the genome that was missing. And even my own institute, and their website at genome.gov often overlooks this fact. And they currently note that the genome was 99% complete in 2003, and there was less than 400 gaps. That's actually not true for very technical reasons that I won't get into. But it's simply not true. There's actually 5 to 10% of the genome that to date has been missing. And the reason people have been willing to kind of overlook this is that the missing parts of the genome don't necessarily contain a whole lot of genes. It's mostly what you might hear referred to as junk DNA or something like this. But it does have important functional roles in the genome. This is showing what's called a karyotype of the 24 human chromosomes, 1 through 22, X and Y. The little bandings are just how it dies when you stain it with a dye. But shown in blue here, I've highlighted the regions of the genome that were essentially unfinished back in 2001. And they localize towards the middle of the chromosome, shown in pink here, otherwise known as the centromeres, where they tend to pinch down together. But yeah, these blue regions have not been finished, and they include some genes. They include important genes called the rRNA genes that code for your ribosomes, which are the machines that crank out proteins. There's also an unknown number of errors and omissions in this reference that was done 20 years ago. So the reason it matters, why we want to go in and improve the quality of this reference genome, is because any variation within those unfinished regions is unexplored. So for instance, if there's a genetic variant that leads to a disease or some other phenotype in those regions, we simply will not have known about it to date, and we won't be able to develop medicines or therapies to address it because we simply haven't sequenced that region of the genome yet. Further, to understand how a genome works, how cell division works, how genome replication works, we need to know the full sequence so that we can design experiments and studies to better understand it. Having gaps in the current reference leads to analysis artifacts, and there might be currently unknown discoveries waiting in these unfinished regions. And it really boils down to a technical reason why it hasn't been finished. This last remaining 5 or 10% of the genome is highly repetitive, meaning it's the same sequence of bases repeated over and over again. So imagine a book where pages and pages just have the word "the" repeated thousands and thousands of times. There's regions in the human genome that look like that, and we call them genomic repeats. And they're very difficult to sequence and assemble because we're not able to sequence the genome in one go. We have to sequence it in little pieces and then stitch those pieces back together like a puzzle. And when all of your puzzle pieces look the same, it's really hard to put the puzzle back together again. And so these repeats have prevented us from finishing the human genome to date, and that has been a technological limitation. And the technology of genome sequencing goes back for 40 years. If you want to go back to the first sequencing paper, we go back to 1975 in a paper by Fred Sanger, shown here in the top right, a multiple Nobel laureate. And the first informatics, the first genome assembly, as we call it, putting a genome back together again, dates back to 1979 from this guy, Roger Staden, who was in the United Kingdom at the time. And here's a couple of sentences out of this paper that's now 40 years old, and it reads like a paper you might see today, that with modern fast sequencing techniques and suitable computer programs, it is now possible to sequence whole genomes without the need of restriction maps. But the sequencing technologies they're talking about here are 40 years old. The computer programs they're talking about here are old Fortran programs, and if you wanted to get access to those, you had to email. Well, I'm sorry, they didn't have email in 1979. You had to write a snail mail to Roger Stadden, and he would put it on cassette tape, basically, and send it back to you. And that's how you would trade your data around 40 years ago on cassette tapes, basically. And then this other quote here points out the key to this genome assembly problem that I'll talk to you about today. And that when you read those individual pieces of DNA, you have to read a long enough piece so that you can distinguish it from being a repeat. So think again about the book analogy. If you read just the word "the," you don't know where in the book it came from. But if you read a sentence of 10 to 20 words, you'll be able to figure out where in the book it came from. So there's our ability to read longer and longer pieces of DNA really influence our ability to assemble these genomes. And so in 1979, they were assembling the short genomes of viruses and phage genomes. And it took really another 20, 30 years until we had the ability to sequence whole human genomes, which are many orders of magnitude larger in size than viral genomes. And it's been within just the past 10 years that we've had a real transformational advance in our ability to sequence longer and longer pieces of DNA. And I'll tell you about two technologies today. The one really, really cool technology that I think should excite a lot of you data scientists is what's called Nanopore sequencing. And there's a schematic of it shown here, where in an amazing feat of engineering and chemical engineering, they're able to have a protein nanopore that's actually taken from the E. coli bacterium, and they puncture a membrane so that they can then feed a single strand of DNA through a bunch of enzymes through this pore. And then they read off with an electrical chip the change in electrical current that's flowing through that pore over time. And you get what's called a squiggle, which is this series of wiggly lines over here on the right-hand side. You can feed those squiggles into machine learning techniques, in particular recurrent neural networks, and decode those squiggles into the A, C, G, and T bases that went through that pore at any particular point in time. And this is how nanopore sequencing reads off a piece of DNA and translates it into computer code as A, C, Gs, and Ts. This has been around for just around 5 to 10 years. And it's really remarkable in its form factor that the sequencers themselves are about the size of a USB drive. So they're very portable. And this is a picture of my collaborator, Matt Luce, sequencing DNA in a hotel room at a conference a few years back, kind of demonstrating the portability of these. The pros of this technology is that because you're able to thread a single strand of DNA, if you can give it very long pieces of DNA, you'll be able to read really, really long reads. And so it has the longest sequencing length, or read length, as we call it, available. And with that, you can get very, very long sentences, like I was talking about on the last slide, that let you resolve these complicated repeats. The downside is that a little bit lower throughput and quality than some of the other techniques, but everything comes with pros and cons. It's amazing that you can read anything with this technique. It's even more amazing that they can now read with 98% accuracy the bases that go through here with this technology and the help of neural networks. Another very cool technology that we've been using lately is from the company called PacBio, called circular consensus sequencing, where the schematic here on the right shows you start with a double-stranded piece of DNA. You adapt little single-strand barbells on the end, and then a DNA polymerase latches on and starts sequencing around this circle multiple times, and it can unzip this double-stranded piece as it goes. And by reading around this circle multiple times, you take an original piece of DNA and you make multiple passes. You read maybe each strand 5 or more times, and then you can call a consensus sequence. So if you made an error on any one pass, the other 5 passes, you'll read the correct base, and you can run statistics to figure out which is the correct base prediction at each location in the read. This cannot read as long of a piece of DNA as nanopore sequencing, but it's really, really accurate, well above 99%, 99.9999% accurate when you put these reads together, and that's because you're reading the same molecule multiple times. That allows you to distinguish different repeats that might be variable by a few different mutations here or there. You'll be able to very precisely tell where that mutation is and what the difference is and how it affects the DNA at that location. So this technology has become very useful for genetic disease diagnostics because you can sequence the genome of your patient and very precisely locate the mutation in the genome that's leading to the particular disease or disorder. So with those new technologies in hand, we became very excited in the last 5 years to go back to the initial product of the Human Genome Project and finish the remaining gaps and the remaining errors that are in the genome. And so if we go back and look at the initial publications in 2000, the Human Genome Project public effort was called the Initial Sequencing and Analysis of the Human Genome. The Celera Genomics effort was called the Sequence of the Human Genome. But even if you look here at the picture, there's not one human genome. We all have a unique human genome, each and every one of us, which is a unique recombination of the genomes of our parents. And so it doesn't make sense to have a single human reference genome. There's no such thing as the human reference genome. There's a human reference genome. And so we started this telomere to telomere consortium with the goal of completing the first human genome, meaning a single human genome end to end. And with that technology and ability in hand, we can go on then to sequence the genomes of any individual, particularly in a healthcare setting as it relates to improving their quality of life. And so 20 years later, we're back here to try to make some revisions to these initial projects, and we want to announce the complete sequence of a human genome. And so my primary partner in this endeavor has been Karen Miga, who is emerging, very promising scientist at the University of Santa Cruz in California. And she has joined up with me in this effort to finish a human genome. And we've built basically a grassroots consortium. We did not have dedicated funding from the NIH outside of our initial groups to do this, but we built a consortium of people that were interested in the genomics of the human genome to complete all of these last remaining gaps. And the first thing that we needed to do is collect a whole lot of those very long reads from technologies like nanopore sequencing. And we don't just need a few of them, we need lots and lots of them. And if you look at a picture here, this is a density plot showing you read length distribution. So if I run my sequencing reaction one time, how long are the reads that I get? You get some reads that are very short, you get some reads that are 30,000 bases long, you get some reads that are 100,000 bases long, and you get some reads that are even much longer, 500,000 to even 1,000,000 bases long. And it's really these very long reads that we were most interested in. And so by producing many sequencing runs, performing many different sequencing reactions, we can collect a lot more of these 500 kilobase or longer reads that make the assembly problem much easier. And so our Nanopore in-house expert at NHGRI, Shalise Brooks, took charge of running this Nanopore instrument called the GridION back in 2018. And Shalise ran this instrument basically nonstop from May to the end of October in 2018, taking many millions and millions of cells from a human cell line that we were using for this project and preparing them and then sequencing on this Nanopore instrument here. In the end, we ended up with about 9 million individual reads, where the average length of those reads was about 100 kilobases in length. So totaling 44 billion bases in total data. And the longest read we were able to achieve was 1 million bases long, a continuous stretch. Compare this 1 million number to just 10 years ago with Illumina sequencing. We were happy to get 100 base pair reads. And we've now scaled up to 1 million base pair reads. And so it's many orders of magnitude improvement in the read length, thanks to this technology. Being a very open consortium and trying to attract other members to look at this data and improve our methods, we performed an open data release about 2 years ago where we put all of this data up on an Amazon S3 bucket in the cloud. From all of these technologies, I don't expect you to know, but I call this the kitchen sink recipe, where we basically throw every sequencing technology that we have at our disposal at the problem and make that data publicly available so that data scientists and algorithm developers can go write new methods to analyze and finish the genome. And to give you an idea of how much improvement this led to over the past 20 years, the initial Human Genome Project was truly an international effort. In today's dollars, it took $5 billion and about 15 to 20 years to finish the project. It was announced at the White House, like I said, a number of years ago. And this was the state of the genome in 2001. Each of these little horizontal black and gray bands are a piece of the genome that had been resolved. Anywhere that you see it switch from black to gray, there's actually a gap in there where they don't know the sequence. And so it's left fragmented. And so you look at this and you see a very fragmented picture where there's a lot of pieces of the genome that have been finished, but they're all very small. If we fast forward 20 years, this is what the assembly of Chalice's data looks like when run through the Canoe assembler, which was developed by Sergey Korin in my lab at NIH. And simply by looking at the reduction and fragmentation from having many, many thousands of little black and gray bars to now just, in some cases, a single color per chromosome, which means we've assembled the whole X chromosome in this case from tip to tip without any gaps, without any ambiguity. And that was really what we were shooting for. And this led to the first human chromosome, the human X chromosome, that we were able to truly finish from end to end and resolve all of the bases in that chromosome for the first time. And this was back in 2019 with the help of this ultra-long Nanopore data. And the way that we were able to succeed in getting across these highly repetitive regions of the genome, such as the centromere, was a method that Karen developed to what I call stitch our way across the genome. And so shown here is a zoom in of the centromere, which is about 3 million bases long in the X chromosome. And each of these individual gray lines numbered here 1 through 12 is a single piece of DNA that was read by the Nanopore instrument. And the little colored lines represent unique variants. So this is a very repetitive piece of the genome. Just like I said earlier, imagine it's the word "the" repeated thousands and thousands of times, but maybe in between a couple of the "thes," there's an "and" or a "hi" or "how are you?" That's what these individual colors are indicating. They're variants from the primary repeat unit. And by labeling those variants on these reads, we can then take 2 reads here, like read number 2 and read number 3, both have a pink and a purple variant. And we can use that to stitch them together. Say these must have come from the same place in the genome because they share these same unique words. And then walk our way from the left to the right side with these very long reads to reconstruct a backbone reconstruction of the centromere. Karen was able to successfully do this. She's a centromere and a satellite DNA expert. In 2020, we announced the first complete sequence of a human chromosome. Since then, our collaborator Glenys Logsdon has repeated that accomplishment for the human chromosome 8, and we now have publications up for both human chromosome X and human chromosome 8 that are revealing some novel biology in how the human centromeres form and function. And the centromeres, for those that don't remember high school biology, are important for cell division, because when you picture a chromosome in that kind of X shape that you always see, the bit that's holding it together in the middle is the centromere, and that's what then is pulled apart during cell division to separate the 2 chromosomes into each of the new cells. So a very functionally important region of the cell, obviously, and it's important for us to understand how the centromere forms and functions. And now that we have the DNA sequence in this region of the genome, we're able to study that in a lot more detail. But both of these projects, chromosome X and chromosome 8, took a long time to finish. Karen and Glenys both worked for a year each for each of those chromosomes. And there's 24 chromosomes that we need to finish. This would have taken another 20 years just to finish the genome. And so we were looking for algorithmic ways, data science ways to speed this process up. And it was really my postdoctoral research fellow, Sergey Nurk, who developed the method that we used over the past year. And it uses what's called a graph, a mathematical construction here. We call these string graphs in genomics and genome assembly. And there's a couple of tricks, data science tricks primarily, that we use to make this work well. The first is what we call homopolymer compression. So DNA is made up of 4 characters, A, C, G, and T. Sometimes a single character will repeat multiple times. Like in this case, we have 4 A's in a row. The sequencers have a hard time reading these repeating nucleotides called homopolymers. And so we take all of the reads that we get and we simply compress down. Anytime we see more than 1 A, we represent it as a single A. And that actually works remarkably well to remove a lot of the sequencing error out of the process. In the end, we'll come back to those and blow them back up to the correct copy number. But to start, it helps to get rid of those. We do a lot of statistical processing to clean the individual sequencing reads and correct them as we can. And then we look for any reads that overlap, just like I was showing you on the stitching slide. Any two reads that share unique variants, we stitch those together in what we call an overlap to build longer and longer pieces of DNA, just like we're putting a jigsaw puzzle together again. And then I'll talk about some of the mathematical approaches we use to then finish the genome. Here's what one of those genome assembly string graphs looks like. I understand that you might have had a presentation from Mike Schatz last week who talked about the genome assembly problem, so hopefully some of you are familiar with this concept. Each of the little colored noodles in this figure represents a piece of DNA that we were able to confidently reconstruct. And we'll put an edge between any of those noodles, just a gray edge. We can't really see here because we're zoomed out so far. But where you see the colors switch, that's where we're uncertain about what comes next in the reconstruction. We have maybe two different options that we can use for our next puzzle piece. We're not sure which is which, so we just leave it unresolved and we connect it with an edge to represent that ambiguity. And so this is what the human genome looked like when we sequenced it with PacBio HiFi and ran it through Sergey's assembly graph method. And each of these components in the graph basically represent an individual human chromosome. I can organize them for you and color them by which chromosome is which here. And you can see this is chromosome X, 1, 2, 3, 4, and so on. But you can also see kind of highlighted in gray here, regions of each of the chromosomes where the repeats live. And that results in this very tangly graph structure where we don't know the correct path. To reconstruct one of these chromosomes from end to end, we need to walk through this graph, visiting each node exactly one time, and go from the left side to the right side. But in cases like this tangle I'm showing you down here on the bottom right, it's just not clear at all how we would walk through that and reconstruct the chromosomes one by one. So we had to look a little bit closer. And another postdoc in my lab, Nico Rautiainen, has developed methods to simplify these graphs and use additional data like the Nanopore data to guide our walks through this initial graph. There's two things I want to show you on this slide. On the left-hand side, in some cases, it's very clear how you need to walk. So remember, each of these noodles is a piece of DNA that we need to visit exactly one time. So we can start here from the yellow and we can visit the red, the green, the red, the green, and then the purple. And if you just look at this for a few seconds, you'll see that that's the only path that goes through this region of the graph that visits each of those nodes exactly once. And so this is called a Hamiltonian path. And you can look at regions of the graph and write an algorithm that will check, is there a single unique Hamiltonian path that traverses from the left side to the right side and visits all of these nodes one time? If it is, that's how you reconstruct that part of the puzzle and you're done. There's other regions here where there's not a way you can walk through this graph and visit each node exactly once. You can either take the right side or the left side. This is caused by what's called heterozygosity or variation in your genome. In fact, every one of your chromosomes is present in two copies, one that you inherited from your mother and one that you inherited from your father. And in places where your mother and father's genome differs, it represents as a variant. And so one half of this bubble will be your father's version, the other half will be your mother's version. And so in these cases, we just pick one. It's not that important for the generation of a reference genome, which of those two we choose. On the right-hand side, it's a much more complicated picture. This is a graph that it would be very difficult to identify a Hamiltonian path through, but in red here is showing a nanopore read that guides our walk through the graph. So we can take an ultra long nanopore read that's a megabase long and we can align it, figuring out which sequences it shares with this graph. And it gives us a reconstruction that we have to visit these nodes in order from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, with this blue one here being another heterozygous bubble like I showed you on the more simple version. So with the combination of this kind of mathematical reasoning and the alignment of long nanopore reads to the more complex graphs, we were able to simplify a lot of these regions and reconstruct the most complex repeats in the genome. Here's just a view of what some of the chromosomes look like, zoomed in a little bit closer. Some of them were easier than others. Here's chromosomes 10, 20, and 8, which as you can see are relatively easy. You could look at this picture for a few minutes and any one of you would be able to find the one unique path that walks through each of these graphs. There's some that are a little trickier. Here's chromosome 2 and 18 that you can see. It just has a much more complex graph structure and a little harder to identify the correct walk. In these cases, we may have needed to bring in the Nanopore data to help guide the reconstructions. And then there's some really horrible cases, the so-called human acrocentric chromosomes, so 13, 14, 15, 21, and 22. Shown here in red in this fluorescence image of the chromosomes is a highly repetitive piece of DNA that occurs in hundreds and hundreds of copies. That makes the reconstruction of these regions of the genome particularly difficult. This is what the graph looks like here. Again, there's no clear path to go from the telomere through this to the centromere and resolve these chromosomes. This is actually representing 4 chromosomes that are all tangled together because of the similarity that they share. So in these cases, we were able, again, to use the Nanopore data and another data type called HiFi to guide our walkthrough here. And I've highlighted here in green the correct path for chromosome 21. So from the end of the chromosome, the telomere, through the middle of this tangle, visiting each of these nodes and out the other end. And we were able to do that for each of the chromosomes tangled together here. And so that took us about a year of looking at this HiFi graph. And we have a consortium of about a little over 100 people. We spent the summer very actively looking at this while we were all in COVID lockdown last year. Each of us was kind of assigned a chromosome, and we worked through these like puzzles, figuring out the correct path through these graphs and looking at the other data that we had on hand to validate and inform those paths. And I'm here to tell you with great joy, we've completed that task, and we have the first complete, truly complete sequence of a human genome that we released in the fall, and we'll have preprints up describing this work in the coming month. And to just give you an idea of the kind of improvement this is over the current reference, here's some basic statistics. Our genome assembly is from this CHM13 cell line. One important note is that we don't have a Y chromosome yet because this is a female cell line. So we only have 23 of the human chromosomes. We're still working on the Y. But all of the unlocalized and unplaced sequences that exist in the current human reference genome listed over here on the left, we've removed all of those. We know where every single base in the genome belongs, and we've placed it correctly. We've added over 100 million bases of new sequence that was never before known in the human genome. We've gotten rid of an equivalent number of gaps, so unknown sequence in the genome. And we've improved the quality by, I would say, an order of magnitude in terms of the accuracy. And so this Q70 number means our accuracy is 99.9999 in terms of quality. So 7 nines worth of accuracy. So that gives us a final human genome size of 3.055 billion bases, so around 3 billion bases for one human genome. And that's an addition of around 3 to 6% of completely new sequence we've never seen before in the human genome, which now we're very eager to analyze to understand its function and any association with disease. Again, following this open data model, we've released this data early in the process. Back in the fall, we briefly announced the data on our GitHub page, and we made it available to the community to analyze. It's the biggest addition of new bases to the human genome in the history of the Human Genome Project. So dating back to 2000, the y-axis here is showing how many bases are in each new version released of the human genome. And you can see it kind of plateaued from the early 2000s through 2010 with very little improvement because of a stagnation in the technology. And then with the advent of the long-read sequencing technology, you can see we shot up and added another few 100 million bases of genomic sequence, culminating in the CHM13 release in the top right here, which was the biggest single version improvement in the genome since 2000. So now our consortium is really moving on to the analysis phase, taking this 200 megabases of new sequence and discovering what's new. Are there new genes? Are there new functions in here that we haven't been able to previously analyze? One of those projects is being carried out by Mitchell Volger, who's a graduate student in computer science at the University of Washington. And he's taking this opportunity to perform an analysis of repetitive DNA across the whole genome. And so what each of these little chromosomes is showing you is regions of duplication where there's been basically a copy paste of a piece of DNA to somewhere else in the genome. And this is a very dynamic and active mechanism of genome evolution, and what might actually describe some of our differentiating factors between us and our great ape relatives, are how these pieces of DNA were copied and pasted around the genome to allow us to evolve bigger brains, opposable thumbs, and all of that good stuff. And so this is basically like a map of the genome now, and it's a complete map of every piece of DNA in the genome, where it's been copied in the genome, and we can relate to how those pieces of DNA differ between individuals. And so as a consortium, we're really taking the methods that we developed here and moving on to bigger and better projects, including what is referred to as the Human Pangenome Reference Consortium. So I alluded to this earlier. Rather than just sequencing one genome, we want to sequence the genome of many individuals. So the Human Pangenome Reference Consortium is undertaking this same project now, not just for one cell line, but for many hundreds of human genomes sampled from a very diverse selection of the human population, trying to get as much diversity as possible. And that's important for one primary reason, that if you go into the doctor of the future and they want to analyze your genome to understand how you might react to a new medicine, what allergies you might have, what your susceptibility for heart disease in the future might be, that information exists in your genome, and we can analyze and understand that. But if we're working with only a single genome, which in the case of the current Human Genome Reference was taken from one volunteer in upstate New York of African American ancestry, if you come into the clinic and want your genome analyzed and you're from a different population group, everything that we know about the genome is based on that one individual. We want personalized genomes, meaning that we know the genome of any one particular individual and be able to give you a personalized prediction of your different genetic risks. So that's the goal of the Human Pangenome Reference Consortium, is to repeat this Human Genome Project now, not just for one individual, but for many individuals. To do that, we're using a lot of the same methods that I described here, but we'll also require new data science, new computer science methods to compare those genomes to one another and understand those differences. And not just for human. In fact, a huge amount of genetic research relies on what are called model organisms. And so these are organisms like zebrafish, Drosophila, fruit flies, mouse, and even non-human primates that scientists use to understand the effect of variations and medicines. So zebrafish, for instance, is a great model for early organismal development. You can have a zebrafish, have it lay eggs, observe the individual baby fish growing up in their eggs, and understand how different variants in their genome affect the developmental pathways and stages. And so improving the genomes of these model organisms is also important to improve research on those species. A related project, which actually was just published today in the journal Nature, is a project that was led by my group, as well as Eric Jarvis at Rockefeller University and collaborators in the UK and the EU, describing 100+ genomes of vertebrate species from across the tree of life, including this beautiful Anna's hummingbird shown here on the cover of Nature today. And we use the same genome assembly methods and sequencing techniques that I described to you today, but applied it to 100 vertebrate genomes. And this is part of what's called the Vertebrate Genomes Project, being led by Eric Jarvis at Rockefeller. And the near-term goal is to go through all of the extant vertebrate species on planet Earth, starting with the taxonomic orders, of which there's about 250, but ultimately progressing to all known vertebrate species, which by our current count is a little over 70,000. And I was explaining this project to my 15-year-old daughter yesterday, and I said, hey, look at this cool cover. We did the hummingbird. She said, okay, that's great, but what do you do with that genome? Like, what's the point of all of this? And that was a very good question. I'm glad she put me on the spot. The point really is to understand the function of the genome. And by looking at all of these different species, it gives us the opportunity to compare their similarities and differences. So for instance, if you want to know how a hummingbird flies, or you want to know why a parrot can learn speech, but other birds can't, you can use the genome to figure out what genes that they have that the other ones don't, what variants that they have that the other ones don't, to understand those differences in the genome and how that leads to these traits, such as vocal learning, which is Eric's primary interest, or flight, or any of these other traits within the vertebrate clade. And so ultimately, it's a journey of curiosity, trying to understand how the genome works to result in this beautiful diversity of life that we have in the vertebrate tree. So I'm going to leave plenty of times for discussion and question. I just want to wrap up with a few summary thoughts on what I've shown you today. The first summary is that the human genome is finally complete. We're 20 years later. We've added the most bases ever. We've filled in all of the remaining gaps. And now we're moving on to the fun of resolving the genes and structures that we've uncovered. This was really driven by technology. In particular, the Pacific Biosciences HiFi technology is extremely powerful in its ability to very accurately read the human genome, as well as the Nanopore ultra-long sequencing. And so what's next, we're going to study the biology of the whole genome for both human and vertebrates. And we're going to move into what I would call personalized genomes for every individual, pangenomes, building a catalog of human genetic diversity, and metagenomes. This is the microbes and viruses that live in and on you and can affect things like your gut health, or different skin disorders, or anything that has a microbial component. We can apply these same sequencing and genetic techniques to understand the effect and influence of those microbes on human health. And lastly, I just want to emphasize again how all of this has been driven by technology. And so hopefully this is a bit inspirational for people that are interested in data science, that we have come so far in the last 40 years in the field of genomics, but that has only been possible because of the equally tremendous advances in technology. And so shown here is the VIC-20 computer, which was out around 1980, and it was the first computer I learned how to program on when I was a kid. And it's orders of magnitude lower computational power than the smartphones that you all carry in your pockets. This is like an old Samsung Galaxy, which has 8 gigabytes of RAM off the shelf, and this old VIC-20 would come for thousands of dollars for just 5 kilobytes of RAM, and 1 megahertz processor versus 2.8 gigahertz processor. So we've gone from like the room-sized computers down to smartphones that are tremendously more capable. Similarly, for DNA sequencing, we used to do these on gels, radioactive gels. They would have to be read out by hand, basically. And now we're able to thread individual pieces of DNA through protein nanopores and decode them with neural networks. Incredible to see the technology that's happened in my career over the past 40 years. And I'm really excited to see what happens next. The future is in your hands as it goes. These future developments will be on the shoulders of folks like you, getting into the field of data science and developing the methods and technologies that will allow us to do things not just in genomics, but over every application. And then finally, just to end with this consortium slide showing all of the collaborators and contributors to this T2T project. It's been a fantastically rewarding effort to work with all these brilliant people, principally my main collaborator, Karen Miga at the University of Santa Cruz, as well as our technology partners. For cloud providers, we've been working with DNA Nexus and Amazon Web Services. And then for genome sequencing technologies, primarily PacBio, Oxford Nanopore, and Arima Genomics. And with that, I'll end there and be happy to have any discussion, answer any questions that you might have about my presentation. Thank you so much.
Yeah, thank you, Adam, for a very informative presentation. Any questions for Adam?
And it doesn't have to be about the talk, career questions, life questions, COVID questions. Anything you have, I'd be happy to answer.
Yeah, so I have one question, maybe career or this. Some of our audience here are graduate students. Maybe some of them are not very in depth with genomics. So if a graduate student wants to venture into genomics, maybe informatics, data science, all of those combined, what do they need to know?
Yeah, it's a good question. And I'll just kind of share with you how I got into it. I mentioned that as an undergrad, I went as a computer science major, and my intention was to do computer science. I chose that as my major because it was something I was interested in that I was good at. And I got into genomics by way of an internship. And so there are a great number of genomicists, or for that matter, any research discipline that is now becoming much more data focused. You have people that were kind of traditionally trained as a laboratory scientist, and now the instruments are producing terabytes of data per day and they don't know how to deal with it. There's a huge demand right now for folks that are trained in managing large data sets, regardless of whether it's genomics, just generally competent at scripting languages, programming languages like R, Python, comfortable working in kind of a Linux, Unix environment, handling big data files, and so forth. And I had no prior experience in genomics, and I was able to get an internship basically as a programmer for a research institute that was developing these kind of genomic technologies. And it was really by way of that internship that I got exposed to these applications, and I was able to apply my programming skills to the kinds of problems that they had in the field of genomics. And so I would say as a data scientist, you know, skills pay the bills, as they would say. So the more programming languages and the more skills you have, the more marketable you will be to folks, not just in the research field, but at places like, you know, the big Googles, the big Facebooks, and so on, and the smaller data-centric companies that need people to analyze and make sense of this data. And that's how I got in, and I think that's how a lot of you all will get in. I really don't expect everybody goes into genomics like I did. You're free to follow your own interests and curiosities, but that fundamental skill set of having a few programming languages under your belt and being able to manipulate large data sets and perform simple kind of statistical analyses and so forth will take you a long way and open a lot of doors into these types of applications.
My next question is, so first, congratulations to your consortium for your effort to complete the sequencing of the human genome.
Thank you.
My question, I mean, they've been talk about personalized medicine over the past couple of, or maybe the past decade. In terms of cost and in terms of what your group have done, how is that, or how would that impact this in using personalized medicine for prediction of clinical outcomes in the future?
Yeah, and that's another great question. I always like to just ignore cost because I'm just trying to do research and get the best products I can, and I often don't talk about how this actually would operate in practice. So let's talk about costs first. As I mentioned, doing the first human genome cost $5 billion. And so obviously that's not amenable to personalized medicine. With 20 years of technology development, the field has driven that cost down to now a few $1,000s. And so if any of you came to me with $5,000, we could figure out a way to get your genome done today. Not a complete genome like I've shown and presented here today, but a genome that would be good enough to get the sequence of all of your genes and look for any potential variants. And this is essentially what companies like 23andMe are doing. When you pay them, maybe, I think now they just charge a few $100s. You send in a sample and they send you a genetic report. It's still the early days. I actually personally haven't gotten any of those services done on my own genome, and I actually have not yet sequenced my own genome because it still is the early days in trying to know and understand what variants in the genome lead to what outcomes and what additional risks. And I don't think that those connections are good enough yet to really change my life in a meaningful way. But I do think within the next 20 years, they will become meaningful as we do more and more human genomes and we analyze the genomes of more and more people with diseases that we're trying to understand and cure. And so it's always a work in progress. The cost now for sequencing a genome is low enough that it can be done for basically every human as part of routine clinical care at a few $100s per genome. The next question is, what's the analysis? Like, what are the killer apps going to be? If I have my genome, what's it going to tell me that informs my future decisions or informs how I'm cared for going forward? And thus far, the biggest wins, so to speak, have been for people that have rare or hereditary genetic diseases, where there's one clear mutation that's the cause of that disease. And by knowing that mutation, now we can develop treatments for it. But those kinds of diseases are relatively rare. It's much more common in the population that we have diseases like heart disease or diabetes, things like this that have very complex causes and can't be traced back to a single gene. But for folks that do have disorders caused by a single gene, we can now identify, in some cases, treat those effectively. And those have been the people that have gained the most from genomics in the near term. In the long term, I think it'll become more widely applicable to everyone in these more complex diseases.
I think there's a question in the chat area if you want to check.
Yeah, how do you fix that variant? It's a good question. So it's not something that's necessarily fixed. In most cases, you use the variant to understand what function of the genome has been broken by that variant. And then that allows you to design or choose a drug to treat that particular disease. And so, for instance, if you have a mutation in a gene that breaks the function of that protein, if you understand that that protein has been affected in that way, you could have a supplement, some small molecule or additional protein can be added to you as a drug that will take the place of that missing protein. There are on the horizon the idea of genetic therapies. So for people that do have genetic disease that's not treatable by typical drugs, there are what's called genome editing technologies where you would potentially be able to go in and correct that mutation in the genome of, if not all of the cells in the human body, the important ones that are associated with that disease. That is, the real space age cutting edge technology. At this point in time, I'm sure people have heard CRISPR discussed in the news, and that's not really on market as a therapy yet. And there's obviously a lot of important safety and ethical considerations that need to be taken into account before that gets rolled out. But that is on the horizon and something that is technically feasible today. I would say, something that's on our mind a lot over the past year with COVID, and I hope all of you have gotten your vaccine, and if not, you're planning to sometime soon. But the Moderna and Pfizer vaccines, for instance, are what are called mRNA vaccines. And that is a tremendous technology that's been made possible only within the last 10 to 20 years, where now we're able to understand the structure of important proteins in the COVID virus. And we're able to give the message to your body, this is how to make this particular protein in an mRNA that gets delivered to your body. Your own cells translate that mRNA into proteins, which then your immune system recognizes, and they get to learn how to practice an immune response on those harmless proteins that your body is now expressing. And that's what gives you your immunity. And so this is a direct application where genomics has directly enabled the creation and the deployment of a vaccine that was, started just last January in terms of its development. It was rolled out, tested, and put into people's arms in under a year, which is just incredible advances. And hats off to the team at NIH and elsewhere that have developed that vaccine that relies heavily on these kind of technologies that I described to you today.
Hi, Adam.
Yes, go ahead.
I had a question about gut health. You're about to talk about it a little bit. Have you gotten more into gut health as far as bad bacteria, good bacteria? How do you measure that? And would that be more of a genetic or environmental kind of issue that affects the body as far as how you eat, what you eat?
Yeah, that's an incredibly good question, and it's a really complicated story. I would say first and foremost, the links between the types of bacteria and viruses that live in your gut and the health outcomes are still tenuous and not well understood. But I think what has become kind of common wisdom in the field is the tremendous potential that's there and recognition of how our genome isn't enough to understand that there's more bacterial cells in your gut than I think there are human cells in your body, if not more, about the same number. And they're producing metabolites and chemicals, and they're helping with your digestion. They're as much part of your body as your own body is. And so there's a huge interest in understanding the impact of that on health. But the links and the causation versus correlation arguments are still not particularly well understood. What else can I say intelligent about that? So what genomics has to offer in the near term is basically telling you what's there. And so the typical approach to gut health with a genomics angle is to use what's called metagenomics, where you would essentially take an oral sample and a stool sample from the individual, and you just take all of that DNA, sequence it, and then that tells you which bacteria are living there and what kind of genes they have present, and also which viruses are living there. And the question now is to take all of those bacteria and associate, are there particular genes that are relating to health? If I give you one easy example, we could talk about antibiotic resistance. And so say you have harmful bacteria, you have an infection, you got food poisoning, and you have salmonella infection. And that salmonella might be resistant to common antibiotics that are used to treat it. We could sequence now the metagenome of your unhealthy gut, determine what antibiotic resistance genes that particular strain of salmonella has, and then use that to choose the correct antibiotic to treat the infection. So that's a real easy, concrete example. As far as the gut health and its relation to, say, obesity or diabetes or inflammatory bowel disease, all of these other gut-related disorders, that is a complex question and a very, very active area of study at the moment. So if you see, there's companies now that spring up that claim, let us profile your gut and we can predict your future health. They're all liars. They can't do that yet. They're selling a promise.
I do see that. Yeah, I do see a lot of that going on.
Yeah, the promise is there, and it's definitely becoming a buzzword. But I would go out spending your hard-earned money on that yet, because they don't have the information they need to make those confident claims yet.
Thank you, and congratulations.
Yeah, thank you.
Any more questions for Adam? Okay, so if you don't have any further questions, Adam, thank you again for a very informative presentation. And
my pleasure.
thank you.
Thank you all for the really good questions at the end.
What was it? I didn't get that.
I just said thank you for all the very good questions at the end.
Oh, okay, all right. And thank you, audience, for coming. I mean, many of you had been with us from the beginning. I can name some names, but I want to acknowledge just one person here, Dr. Akala. He is a professor at Howard University, one of the leading researchers, and then he's been with us from day one. So Professor Akala, thank you so much for your support with this training series, and thank you all for coming. Hopefully, if we have to do this again, you will all participate, or you will at least recommend the training to your colleagues, students, or friends, whatever that may be. So do well to complete the survey, and then those of you who would want the digital certificate, I think you will have to send us a note, and then we will send you the certificate. Other than that, if any one of you have any questions or anything, we can discuss that. This is the last day, so this is the opportunity for us to do so. Any
thank you, John. Thank you, everyone.
or anything?
Yes, so I think one of the questions that comes up, thanks a lot, Adam. I'm sorry I missed most of your presentation, but I'll go back and look through the recording. Excellent. One of the questions that do come up very often is whether Codio will be available. And so if Nana can address, I think my intuition is that it's not going to be, but if Nana could address that, and if people want to what will happen to the files that are currently available in Codio?
Yes, so Codio will still be available for the users to use it. So yes, it will still be available. So have your login, you still have the files there. GitHub will still be available for the resources. And even though we are still officially, the session is over for VADSTI, you can still reach out to the Slack for any questions, and probably we can make time for you for some extra TA sessions if they need to be. So yes, Codio will be available, GitHub will be available, and Slack will still be available. Yeah.
Thanks, Nana.
So
And
yeah,
one thing
look out for VADSTI 2.0 coming up very soon. So we have some surprises coming. And is there a survey link out there, John?
Yeah, I think Stacy posted that. Yeah, so it's there.
Yeah, so please do well to complete the survey and let us know some of the areas that you want us to cover and all that kind of stuff, the ideas that you have, and we'll try and provide our resources to you.
Yeah, Nana, I think there's a question from Dr. Omari. He wanted to know how long is it going to be forever, or is there going to be a time limit?
For which one?
I think for all three. GitHub, I think, is going to be.
GitHub is yeah. GitHub will still be around.
Okay.
I mean, it will still be around. So like I'm saying, Slack is there. So just send all your information to the Slack, and also do well to recommend your other colleagues about VADSTI, and let us get more people on board and make this happen.
And then, yeah, John, I think there's a question in the chat. Will we get an email about VADSTI 2.0? Is the group going to be informed about 2.0 and what additions?
Yes.
I know you don't want to give out. Yeah.
Yes, we'll do that. Not only VADSTI, if there are any other activities coming up. For example, there is a Codiothon on health disparities that will be in, I believe, in May. You can check Slack, and then if you find it interesting, yeah, you can sign up for those. So we'll be doing that, not only VADSTI 2, but any activities going on that we feel you may find it useful, we'll send that to you.
Okay.
Yes, when will we be given the certificate? Yeah, I think once you meet the requirement, the certificate will be sent to you. Any TA session this weekend? William, you want to answer that? I think that the session is officially ended, but William, what do you have to say on that?
Yeah, so because participants have been very, they've sacrificed a lot, so we'll throw in one more weekend of TA session. I can't speak for yeah, I can't speak for Ibel, but yes, Saturday 9 a.m. and Sunday 5 p.m. So 5 to 6. Normally we say 5 to 6, but if there's a need, we normally drive on. And Saturday 9 a.m. normally drives on to about 11. So please join us if you have any questions. And then Ibel, I think Ibel also has something, but I can't speak for Ibel. I'll let him speak for himself.
Stacy, if you are there, can you answer the question about the certificate and what they will need to do?
I'm sorry, could you repeat the question?
There is a question from Helen on how will we get the certificate and what do we need to do?
Certificates will come out most likely next week. There's nothing that you need to do on your end. Certificates will be sent out to people who have attended at least 80% of the sessions. And if they weren't able to attend the session, at least attended the TA session, but we will send that out to everybody individually, most likely starting next week.
Okay, so if there are no further questions, again, thank you very much for making this training a success, because without your coming, this wouldn't be successful. So we thank you, and again, look out for any notices or announcement in Slack about other upcoming activities, and then thank you again, and all of you have a good day, and then enjoy your weekends. Well, today is not Friday. I thought today was Friday, but it's okay. Still enjoy your weekend whenever it comes. All right. Bye, everyone.